I'm sure he's known to many of you who have dialed into this webinar. So Bob began his career in industry with Seneca, and for the last 12 years he's been with Pfizer in Sandwich, where he's a senior director and head of material sciences. So Bob will give us a presentation on the impact of solid form informatics in the pharmaceutical industry. I think he has a unique perspective on this, and is really a key opinion leader in this area. So over to Bob.
Thank you, Colin. Good afternoon, everyone. What I'd like to do this afternoon is take you through the bridging of some of the new emerging computational methods to industry practices to facilitate and enable solid form and particle design. And really this transition from being a function that actually screens for looking for solid form to an organisation that actually tries to design the solid form for moving a candidate forward at speed. So in the presentation, what I'm going to do is describe the importance of material sciences and solid form selection on product design, give a personal perspective on the evolution of material sciences over the last 15 years, and highlight some scientific advances in academia around solid form informatics, polymorph, prediction and particle engineering, and describe those in the context of trying to design a commercial solid form for robust product design. We're going to talk a little bit about how these can be applied during drug development, future perspective and acknowledgement. So, in the first slide I just wanted to overview some of the challenges that the industry faces. This is a generic attrition curve that we're seeing here. So in discovery we screen many compounds, and eventually that filters through to a number of key products in development and through to registration. But there are some challenges for the industry. Firstly, the industry is not getting any faster. We are making less API and drug product batches. So in essence we are moving from a philosophy of testing ourselves into compliance to actually being in a place where we're actually building quality in during the design phases of the product. Molecular complexity is increasing, and with that comes a solid form complexity that we have to try and address. And there are increasing expectations from the patient on the product performance, and rightly so. And what we're talking about, I think, is the journey. Pharmaceutical sciences really enables the journey from the molecule to the product, the molecule to the medicine. And during that transition, we need to be able to design API manufacturing routes that isolate the correct solid form, and we need to be able to have analytical testing to support that, and then we need to make sure that that solid form is suitable for processing through to the final drug product. And this solid form and particle nomination is really a key milestone in the design of the commercial product. And you can see as we go through this journey, molecule to the crystal to the particle, and the particle to the product. And what we need to understand on that journey is the impact of the free form, the salts and the co-crystals, preferably avoiding hydrates and solvates as they tend to complicate the solid form space. But understanding the solid form key attributes, the chemical stability, the physical stability, the solubility and bioavailability, and the bulk processing. And really by understanding the link between the molecule and the crystal and the product, we're able to underwrite a quality by design product and delivering safety, efficacy and manufacturability. And you can see on this slide here, pharmaceutical material sciences, quality by design, the importance of manipulating the solid state chemistry to achieve various attributes, whether it's the efficacy, so choosing the right salt or co-crystals, understanding the biopharmaceutics, making sure we isolate the right polymorph, through understanding the impact of the solid state chemistry on dissolution, on the shelf life of the product, the chemical stability, the homogeneity of the product, the evenness by which the API is distributed in the product, how easy it is to process the API through to drug product, and indeed how easy it is to isolate the final API through the crystallization process. And what you can see is that solid state chemistry being a foundation element of the delivery of the product, the quality of the product, and the manufacturing efficiency of the product. So really this journey that we've been on, this journey to produce a greater structural definition of that journey from molecule to medicine, has really been shaped by academia. And this slide is a build slide, and what you'll see as we go through this, the influence of academic invention in shaping the way we think about product design in the pharmaceutical industry. So clearly we have the Nobel Prize for supramolecular assembly. That then resulted in people starting to think about crystal engineering, the design of organic solids. You can see throughout the literature a number of examples where the influence of that thinking is starting to permeate its way through both chemistry and material sciences. You start to think about not just the design of organic solids, but the design and application of functional solids. And ultimately, you end up with an MRS bulletin review where you start to introduce the concept of pharmaceutical material sciences. And this idea of being able to engineer the product from the molecular structure upwards. This transition of molecule to crystal, crystal to particle, and particle to product. And how has that evolution in academia actually shaped the changing perspective of material sciences in the pharmaceutical industry? When I first started, what we did, I think, was deliver relatively routine characterization of drug substance and drug product batches. We ensured the quality of what was being generated. What we did then, I think, was invest in better characterization, whether it's by powder X-ray diffraction, single crystal X-ray diffraction, thermal methods. We then started to think about automation and robotics to allow us to search for different solid forms and indeed introduce intellectual protection associated with it. We then started on a journey that said, if we've got the solid form, how can we check its suitability for manufacturing? So we introduced these small-scale material testing, whether it's check the compactability of the API, the flow of the API. And increasingly we've started to look at surfaces, and in particular use of techniques like AFM, to examine the surface attributes of an API, particularly the importance of that with relation to inhalation products. But ultimately I think we're on a journey that takes us towards a structural perspective of solid form selection with the integrated application of computational tools towards solid form and particle design. So the use of the sort of techniques and tools you hear in the 4 other webinars that are part of this process, describing some of the opportunities that are emerging to describe the solid form and particle space that may be accessible to scientists. So let's think about how we build on experience. And this really shaped a lot of our thinking. There's 2 fantastic papers published by our colleagues in Abbott that describe some of the challenges associated with Norvia. I think what's interesting as you go through those papers, you see the learning opportunity for the industry, for us as a community. And in particular, the 3rd bullet point describes the paragraph that I thought was very powerful in the publication. And it talks a little bit about the hydrogen bonding networks that are available. So this second polymorph of Norvia had a stronger hydrogen bond on receptor connection And since the strength and completeness of our hydrogen bonding was obtained the maximum possible in the Form 2 lattice, it wasn't thought possible that another undiscovered polymorphism already ever exists with equivalent or lower solubility. Two fantastic papers that are really worth reading through in some detail. So what that informed us of is a journey that said post a polymorph issue, where we have the tools and techniques that allow us to retrospectively fix and respond as an industry to the challenge. It wouldn't it be wonderful if as a community we were able to actually explore and understand the solid form space proactively before the nomination of the commercial form in the product. And if you go to the next slide, what you'll see is a very early example of some work that we did within Pfizer, looking at a candidate as it was emerging from discovery. What we looked at was from the crystal structure, we started to look at the hydrogen bonding motif. We looked and said, is that a likely hydrogen bonding motif? Has it got the right distances? We then went into the Cambridge crystallographic database and started to examine those interactions using the tools that were available. It was very much done by hand. It was done by me, a non-expert, and trying to look at distances and motifs and articulate the risk of there being a different polymorph out there. What we found was it was quite uncommon in terms of its motifs, and the distances were quite long. That meant we targeted some experiments to try and find the single hydrogen bond shown on the right-hand side, and indeed with a few targeted crystallizations, we did in fact find that. So that was done by a non-expert, and what I think we've got in the next slide is really thinking about, can we make that more accessible to the community? If we make the technology accessible to the formulation scientists and the process development scientists. And really what we're talking about is a journey from a fantastic paper around encoding and decoding hydrogen bond patterns by Margaret Etter, through this chemical communication to celebrate the 40th anniversary of the Cambridge Crystallographic Data Centre around crystal structures and drug design, through to knowledge-based approaches to crystal design, again as part of the 40th anniversary, all the way through to actually developing tools and techniques that allow us to examine hydrogen bonding patterns we might expect to see based on the molecular structure of the candidate we have and the wealth of information that's available in the Cambridge Crystallographic Database, 500,000 compounds that are in there. And actually these tools and techniques are now routinely used within Pfizer to examine all the candidates coming forward to look at what are the hydrogen bonding motifs we might expect to see and compare and contrast them against the crystal structures that we have. So in the second set of examples, I want to move on from an informatics base to actually looking at crystal structure prediction. Whereas in the previous example we were using the wealth of information in the database, in this example we're actually going to try and use that information plus computational tools allow us to examine all possible packing motifs and describe the intermolecular interactions between those motifs and rank them in terms of solid state stability, effectively the last anything. And of course what you're able to take the molecular structure, stick it in a computer, come up with different packing motifs, and ultimately what we want to be able to do is predict physical properties, mechanical properties, crystal shape, and ultimately the likelihood of other polymorphs appearing. And this journey from molecule to crystal in the computer requires a number of things. You require an understanding of the conformation that you're likely to see, or conformations. You're going to need to understand and describe the intermolecular interactions between the molecules. And ultimately you're going to have to be able to search all possible space for different packing motifs. So what you can see is this journey of molecules, the accurate three-dimensional molecular geometry, a whole host of possible packing motifs, and then a method of assessing those in terms of relative stability. And I think over the last 5 or 6 years, huge progress has been made by a number of groups around the world, and in particular there's the blind test run by the CCDC that starts to show the power of these technologies to help understand solid form space as you're progressing a candidate forward. And we have a simple example here in Pfizer where we've actually used these tools and techniques to look at transformational flexibility in candidates moving forward. We were actually worried about a particular candidate being in the more stable solid form. We did some experiments. We also ran some confirmation analysis, and we also ran some polymorph prediction work. And clearly you can see the density total energy maps that are generated by these techniques. We found the possibility of a more stable form, and then when we changed the solvents that we're using for our polymorph screening, we were able to identify this second form B that was more stable. We're really using computational tools to inform and guide our experimentation. The third example, in terms of the application we're going to talk about, is actually starting to move from the crystal out towards broader practical attributes. When you solve the crystal structure, you're able to see the packing of the molecules within the crystal, but of course you're actually able to link that to your external morphology through the Miller indices. The Miller indices, as described on the crystal shape here, allow you to actually link your surface chemistry back to your bulk attributes. And so from the crystal structure, we're able to go and look at what the crystal structure, what the surfaces of the crystals are, and what the possible chemistries are on these surfaces. It leads you to understand, I think, the impact of solvent and habit modification. Allows you to understand the potential impact of impurities on crystal growth. Can often be important when you're trying to develop a commercial API synthesis. And also start to look at things like the surface of the API and how it interacts with the surface of excipients or other APIs. So you start to open up this area of manipulating the environment to produce the crystal habits that you want. And here we have a couple of examples of the sort of technologies that I'm talking about. So this is glutamic acid. What you can do is you can take the crystal structure of both polymorphs. You can predict the external crystal shape based on surface energies and attachment energies. You can actually then look at the chemistry pointing out of these different crystal surfaces. And you can in fact start to look at the impact of changes to bulk to surface ratios and understand the impact of those on relative dissolution rates. So really this journey now out from beyond the chemistry to the surfaces and how the API interacts with its environment and with other parts of the product is a really important part of the journey we're on in terms of designing products in a quality by design philosophy. We can actually go beyond just looking at single particles, but start to look at interparticle binding. So you can start to look at where are the chemical motifs that will fuse together and form the best interparticle interactions. So here you can see again two glutamic acid morphologies and starting to pull together simulations that allow you to look at these two particles coming together and what are the most likely interactions, right? So extending this concept of syntons from just within the crystal to include extrinsic particle-particle interactions. You can see a wealth of innovation or invention happening in academia that we should perhaps consider fusing back into our industrial processes. What I've tried to give you in those examples is a sense that we're starting to view the world between the API and the drug product differently. We're starting to think about material sciences as the discipline that connects those two threads of product development. So if we have an API synthesis, commercial synthesis being developed, and we have a commercial drug product being developed at the same time, what we need to be able to do is fuse those two work streams. And really material sciences through particle characterization, solid state characterization, surface characterization, and indeed solid form design, is actually providing the connection between those two work streams. And really, that connection is critical to the quality of the product that you design. And structural tools, I think, are emerging as the storyteller, the thing that links characterization techniques and allows you to tell the story from a molecular perspective up to a holistic product design perspective. So when would you use these tools? I think this is one of the areas that as a community we probably want to consider. As you go through this attrition funnel, if we're working early with medicinal chemists actually trying to shape the crystallizability of some early molecules, what we want is some fast tools that are intuitive, easy to use in partnership with the medicinal chemist. As we emerge through that funnel and phase 1 and phase 2 exploratory development, we actually want to start thinking a little bit more about the tools that solid form selection scientists are going to use to guide the experiments and drive them in the right direction. So we're actually using the tools now to inform and guide experimentation. And there is a huge opportunity, I think, as we enter phase 3 in tool development, to really think about how can we validate the tools, how can we validate some of these computational methods so as we can describe them in a regulatory dossier in a way that gives the regulators confidence that we may have done that experiment, but we actually understand our solid form design phase and product design phase better than we've done before, and that will then inform our control strategy appropriately. So what does the future hold? Well, for me it holds a structural perspective, a structural definition, of a digital definition of product design. The components of that are the ability to describe the molecule in an intuitive way that's easy for process chemists, formulators, medicinal chemists to understand. It's about being able to describe the solid state in ways that are accessible to the broader community, not just the crystallographic community, but to your formulation, your process development scientists, and your analysts. Being able to describe that solid state structure much more intuitively to that community. I also like to be able to calculate properties and analytical signals. Here we've simulated a PXRD pattern to illustrate the point. So to be able to calculate both properties and simulate some key characteristics of the API. And ultimately moving up to that particle and surface understanding. So being able to describe the different chemistries that exist on the different faces of your API and how that will interact with your crystallization solvent, with your impurities, and with your excipients as you design your product. And ultimately, I think the thesis is that if you think about this journey from API, molecule, through to product, the solid form that you select, how you choose to form that, will ultimately impact on the quality of your product. So the greater your consistency in your assembly of your API into your final physical form, the greater the consistency of your product. So this form, formation and formulation, as we go on the journey from the left-hand side of molecule through particle, molecule through crystal to particle to actual product. As we understand that journey more effectively and put that structural definition in there, I think we will build and fuse the connections between the API internal structure, the solid state chemistry, the link back to the final step of the API, and the link forward to the formulation design. What I'd like to finish on is some acknowledgements to key scientists who helped contribute to this presentation, both within Pfizer, here at Cambridge, and of course at Leeds. And whilst I've given you a view of some emerging tools, I would like you to think of them from a very practical perspective. If I'm a development scientist, the sort of questions I'm interested in understanding is, How often do these new forms, these new polymorphs appear? Will they appear for my candidate? How many experiments do I need to do to ensure robustness? How complicated is my solid form state? How much do I need to explore? And what will be the impact of any changes on my solid state structure from the biopharmaceutics and my stability? So can I describe that in a more holistic way? From a regulatory perspective, I'm sure two questions. The probability of new polymorphs appearing and the impact of those new polymorphs. So those are the two key decision points of ICH 268. And what I've hopefully done in this presentation, and I'll try and answer your questions as well, is take you through some emerging tools. And I believe if we harness these new capabilities to our small-scale material testing, I think that leads to a creation of design by first intent to actually design the API properties for the product, and that is the next evolution of material sciences. So I'd like to thank the scientists on the final slide for their contribution, and thank you for your time this afternoon.
Okay, thanks Bob for a really nice talk there. I do have a few questions that have already come in. Although during this session, if anyone has any more questions that they want to type into the chat box, send to the panelists, then that would be great, and we'll try and include a few more. So to begin with, here's the first question. Computational tools have traditionally been well embraced by discovery chemists